This map highlights the primary endemic zones for the Chandipura virus, focusing heavily on central and western India. The pathogen is a member of the Rhabdoviridae family, specifically the vesiculovirus genus. It carries a negative sense single-stranded RNA genome inside its spiked, bullet-shaped envelope. This specific virus aggressively targets pediatric populations. In children, it triggers acute encephalitis syndrome, a rapid, aggressive inflammation of the brain. The clinical manifestation is immediate and severe, demanding high-level critical care and mechanical respiratory support as the central nervous system deteriorates. Looking at this chart of the 2024 outbreak in Gujarat, we see 245 suspected AES cases. Of those, 64 were laboratory-confirmed as Chandipura infections, carrying a severe 33% case fatality rate. These numbers align closely with historical baselines. During the 2003 outbreak in Andhra Pradesh, fatality rates also soared, proving that this pathogen maintains a highly lethal, unresolved pattern. The data confirms that Chandipura is a recurring pediatric threat. Mitigating its impact requires absolute clinical precision and rapid triage. Institutions like the ICMR, National Institute of Virology in Pune, are actively mapping the virus's recent surge. Their research reveals a stark divide. While our clinical understanding of how Chandipura kills a host is highly detailed, our epidemiological understanding of how it survives in the wild remains largely incomplete. Because we lack a complete map of its ecological life cycle, massive populations are left exposed to unpredictable, recurrent outbreaks. The entire clinical timeline begins the moment an infected arthropod pierces human skin to take a blood meal. Tracking this on a 72-hour timeline, by hour 24, the patient presents with high fever and severe vomiting. Crossing the 48-hour mark brings acute neurological impairment, intense seizures, and altered mental state. By hour 72, unmanaged brain swelling rapidly causes coma or death. To understand why this deterioration happens so quickly, we have to look at the microscopic mechanisms inside the brain. The Chandipura virus is a highly neurotropic pathogen. It rapidly breaches the central nervous system to invade neuronal cells. Once inside the neuron, the virus activates a specific cellular trigger known as the FAS-associated death domain, or FAD pathway. Triggering the FAD pathway activates enzymes called caspase-8 and caspase-3. Together, these force the neuron to undergo irreversible apoptosis, programmed cell death. This targeted cellular self-destruction spreads, causing massive, unrecoverable structural damage to the brain within a matter of hours. The velocity of this viral attack dictates the entire clinical challenge. The therapeutic window is exceptionally narrow, meaning doctors cannot afford to wait for standard diagnostic delays. When a patient arrives deteriorating by the hour, clinicians must immediately select the correct diagnostic modality to confirm the virus. For diagnostics, we have molecular RT-PCR on the left and serological IgM ELISA on the right. ELISA detects host antibodies, but overlaying the 72-hour timeline reveals a fatal flaw. The patient reaches terminal failure before antibodies are detectable. RT-PCR bypasses immunity entirely. This method offers extreme sensitivity. It can detect viral loads as low as 10 plaque-forming units per milliliter from acute blood or cerebrospinal fluid samples taken immediately upon admission. A third option, live viral isolation and culture, does exist and is highly accurate, but it carries strict limitations for active clinical use. Propagating live Chandipura requires biosafety level 3 containment facilities, which are rarely available in the rural clinical settings where outbreaks typically occur. Rapid neuronal apoptosis will not wait for adaptive immunity to kick in. Molecular diagnostics like RT-PCR are the only reliable tool for confirming the virus in time to intervene. We know how the virus kills the host. The epidemiological blind spot is how it sustains itself in the environment for years between these massive outbreaks. The primary biological suspect for the transmission of this virus is the phlebotomine sandfly. Historically, researchers blamed phlebotomous species. However, recent entomological data shows that Sergentomyia sandflies now dominate the exact zones where outbreaks occur. This presents a paradox. Standard vector-borne pathogens require a continuous transmission cycle, bouncing between insects and a mammalian reservoir host to survive year-round. Chandipura skips the mammal entirely through vertical transmission, The virus passes directly from an infected female sandfly into her eggs, hatching an infected generation. Furthermore, adult sandflies can pass the virus venereally during mating, spreading it laterally without a blood meal. Together, these mechanisms create a silent inter-epidemic reservoir. The virus survives indefinitely across successive insect generations, remaining totally hidden from public health surveillance. This biological reality severely complicates traditional vector control measures, especially in endemic agricultural zones, where sandflies breed rapidly in soil and animal burrows. Chandipura bypasses standard transmission models by using the sandfly as its primary amplification reservoir, allowing the virus to persist in the soil until the next monsoon season. This silent survival tactic introduces a final variable that expands the threat profile far beyond India's borders. Looking at the African continent, genomic surveillance has detected Chandipura RNA inside sandfly populations in Senegal, Nigeria, and Kenya. Yet despite the virus circulating in African vectors, there are no documented human outbreaks in these regions. This points to either massive silent circulation or undefined genetic mechanisms restricting the virus from infecting local hosts. This leaves us with a stark matrix of realities and gaps. We understand the rapid 72-hour apoptosis pathway, and we know RT-PCR is the only viable diagnostic tool. But we still lack a confirmed mammalian reservoir and have zero targeted antiviral therapies. The immediate scientific imperatives are clear. Researchers must establish definitive vector competence in the lab and engineer targeted therapeutics that can outpace the virus's accelerated attack on the nervous system. Simultaneously, rapid molecular testing capacity must be expanded deeply into the rural, endemic zones where outbreaks actually hit. Until the scientific community bridges these epidemiological gaps and maps the true reservoir, public health systems are forced into a reactionary stance. They can only treat the aftermath of a 72-hour countdown, leaving genuine prevention out of reach.